This is part 2 of the Chapter 13 lecture notes. We left off with slide number 11 on genomic imprinting, which is a form of epigenetic inheritance. I get my highlighter going here, where either the mother or father's gene found in the egg or the sperm, not both, but one of them, has a methyl group. And a methyl group is one carbon and 3 hydrogens attached. So it's like CH3 is what it looks like. But when a methyl group is attached to the gene, a lot of times what that does is it causes it not to be expressed. And this could explain differences in gene expression in identical twins. Because, as you know, identical twins have identical genomes. So their genetics would be identical. If a DNA fingerprint test were to be done, they would test as the same person. There would be no differences in their actual DNA. So what can explain the differences in the little differences that you notice in identical twins, especially the older that they get, could be this whole idea of genomic imprinting, where either the mother or father's gene has a methyl group attached. So, okay, moving on to transcriptional control. A reminder from Chapter 12, transcription is when DNA codes for messenger RNA, and this happens in the nucleus. It begins when RNA polymerase binds to a promoter region of the DNA. So when RNA polymerase binds to the promoter, that is when transcription begins. Transcription factors are proteins that assist the RNA polymerase binding to a promoter. So in that way, they control transcription. So transcription factors assist RNA polymerase binding to the promoter. So now we find out that for transcription to occur, it's a little more complicated than what we learned in Chapter 12. It's not just RNA polymerase binding to the promoter region of the DNA. It is also this incorporation of transcription factors. And often, further down from the promoter on the DNA molecule, there'll be a region called the enhancer. The enhancer is another piece of DNA, just like the promoter, but there will be transcription activators that will need to bind to that enhancer region, and sometimes even modifier proteins will bind to those activators. And then when they attach to the transcription factors and the promoter, they form a loop in the DNA. It's like a bobby pin or a hairpin. So we call it a hairpin loop. So they form a loop as they attach to the transcription factors and the promoter and the RNA polymerase on a different part of the DNA molecule. And you can see it here. It's very difficult to explain, but here's the promoter region that the RNA polymerase binds to. In order for transcription to actually begin, the enhancer region of the DNA has to come into play. There's these proteins that have to bind to both the enhancer and the promoter, and it kind of bends this portion in here. It bends it into this hairpin loop. So here's your enhancer region, here's the loop, and then here is the promoter region. Okay, so that's the hairpin loop there, and what you see happening is the transcription activator is binding to the enhancer. So here's the activator binding to the enhancer. Furthermore, mediator proteins, I called them modifier proteins in that other, I'm so sorry. That's completely my fault. I don't do all of these slides, but I did that one, and I need to fix it. This should be mediator, and I will change it in just a minute. Mediator proteins. In fact, I can't deal with that. I'm going to change it right now. Engine show keep. All right, we want to come here and do mediators. Mediators can bind, okay? And so let's go back to our picture. So the mediators are these half moon shaped structures, and they bind to the activator. So you've got enhancer, activator, mediators, and all of that binds to the transcription factor complex that is bound with RNA polymerase and the promoter. So I know that's a lot, but the whole point is that transcription cannot occur until this entire complex forms between the enhancer region and the promoter region. And what this picture doesn't show is transcription activators will cause transcription to eventually happen once all of the proteins are bound in place and the complex has been formed and the hairpin loop has been formed. The activators will actually lead to transcription. But if this was not an activator, if it was a repressor would cause it to not happen. So that's one example of transcriptional control, which is controlled by these transcription factors. Now, post-transcriptional control, this is after the messenger RNA, has been produced. The primary messenger RNA transcript, this is before any of the alternative or alternate messenger RNA splicing, what we call messenger RNA processing. So this will affect which introns are cut out, which exons are spliced together, and it can also control the speed of the messenger RNA leaving the nucleus. And any of that is going to control how much protein product gets made at a time, so the rate that the protein product gets made. And this is showing you an example of alternative processing of the pre-messenger RNA. Back here it was called the primary messenger RNA transcript, same thing as pre. So in one example you get, as the introns are cut out, you get the exons A, B, C, D, and E are a part of the final messenger RNA transcript, and that creates a certain protein product 1. But if alternative splicing happens and you only get A, B, D, and E, that's going to be a different messenger RNA transcript, and it's going to produce a slightly different protein product. So this is, again, this is post-transcriptional control when you get alternative processing of the pre-messenger RNA. Now, sRNA is stands for small RNA. So there's a lot of DNA in our entire genome. There's a lot of DNA that doesn't code for anything. We call it non-coding. So the non-coding DNA is used to code for sRNA molecules or small RNA molecules. And small RNA molecules can be involved in regulating transcription and translation. So it just depends. Small RNA molecules are the source of microRNAs, which cause the destruction of messenger RNA before they can be translated into a protein. And they're the source of siRNAs, which stands for small interfering RNAs. that prevent the expression of messenger RNA as well. And either one of these, anytime this happens with the small interfering RNAs, where they interfere with the expression of the messenger RNA, we just call it RNA interference. So small RNA molecules are the source of microRNAs and small interfering RNAs, and both of these will regulate the breakdown or destruction of messenger RNA. So it would prevent a protein from being produced. And then this kind of shows you, all right, starting with step 1, here's the DNA. And some of that DNA we call the non-coding region of the DNA can code for RNA, and it can this particular RNA has formed a loop and actually come back on itself with 2 complementary strands. It almost resembles DNA, except it's not twisted. It's not a double helix shape, but it's a double-stranded RNA molecule. And the next thing that happens is it gets broken up, and those small pieces are your sRNAs, your small RNAs. And they can either form into microRNAs, which will lead to the breakdown of messenger RNA, or they can form into small interfering RNA molecules, which will lead to the degradation of messenger RNA before it even leaves the nucleus. The microRNAs will allow the messenger RNA to leave the nucleus, but still prevent translation, so prevent the protein product from being produced. Then we have translational control, and that determines the degree to which messenger RNA is translated into a protein product. And it basically depends on the presence of the 5' cap, which, remember, the 5' cap is composed of a modified guanine, or G, nucleotide, and the length of the poly A tail. Remember, it can be from 150 to 200 adenines, a chain of hundreds of adenine nucleotides on the 3' end of the messenger RNA. And that cap and that tail are very important in determining how long the messenger RNA remains active. All right, post-translational control affects the activity of the protein product. So post-translational control doesn't occur until the protein is actually made. One of the chemicals that can control the protein after it's made is an enzyme called a protease, because proteases are enzymes that break down proteins. So even after the protein gets produced, proteases can break it down. So what they do is they regulate how long the protein remains active in the cell. And proteases are dangerous unless they are protected in a vesicle, either a vesicle called a lysosome or a proteasome, because otherwise they would break down things that they shouldn't break down, useful things, that are needed by the cell. And this just shows you a protein product that has been produced, is tagged with these green molecules here, and it enters the proteasome, which contains proteases, which break up that protein. And section 3 is on gene mutations. A gene mutation is defined as a permanent change in the sequence of bases in DNA. So it's just a change in the bases of the DNA. Doesn't necessarily mean good or bad. If the mutation occurs in sex cells, we call it a germline mutation, and it can possibly affect the offspring if those sex cells, like the sperm or the egg, actually fertilize, actually undergo fertilization and form a zygote, and that zygote grows and matures and actually gets born. then the germline mutations can affect the offspring. But somatic mutations occur in body cells, and they will not affect the offspring of the individual. All right,